Introduction. DNA has two important functions in the cell. It is used for replication of genetic material in dividing cells. It carries the information for protein synthesis in all cells. Overview of replication. When cells divide, each daughter cell has an exact copy of the genetic material. Copying of DNA is called replication. It occurs in every cell before cell division occurs. Replication. DNA unwinds when an enzyme called helicase causes the hydrogen bonds holding the two DNA strands to break. The two halves of the molecule separate. The two strands are held apart by DNA-binding proteins. Nitrogen-containing bases. DNA contains 4 different bases called adenine, guanine, cytosine and thymine. Adenine, A, and guanine, G, are called purines as they have 2 rings within their structure. Cytosine, C, and thymine, T, are pyrimidines as they have 1 ring within their structure. Finally, all the nucleotides are joined together by DNA polymerase and DNA ligase. 2 new strands of DNA are formed which still retain half of the parent DNA. This method of replication, where 1 strand is maintained, is called semi-conservative. Something to think about. Human DNA is replicated at about 5,000 bases per minute, and DNA can be up to 6 times 10 to the power of 9 base pairs long. Why, therefore, does it only take a few hours for our DNA to replicate? The original DNA double helix unwinds to form multiple replication forks at the same time. This means that sections of DNA are actually replicated simultaneously to make the process much quicker. Evidence of the semi-conservative method of replication. Mendelssohn and Stahl showed that replication occurred by a semi-conservative method. 1. They cultured E. coli for many generations on a medium containing a heavy isotope of nitrogen. When this DNA was extracted and centrifuged in cesium chloride, a region of heavy DNA was observed. 2. The E. coli were then transferred to a culture medium containing nitrogen-14. After 1 generation, bacterial DNA had a lower density as it was made up of 1 strand of the original nitrogen-15 and 1 new strand containing nitrogen-14. 3. At each succeeding generation, there was found to be less nitrogen-15 DNA and more nitrogen-14 DNA. Something to think about. What would Mendelssohn and Stahl's results have looked like if DNA was replicated by a conservative mechanism? After 1 generation of bacterial growth, there would be equal amounts of heavy and light DNA. Following each subsequent generation, there would be increasing amounts of light DNA and still the original amount of heavy DNA. Overview of protein synthesis. Protein synthesis involves 2 stages. 1. DNA is transcribed to form messenger RNA. 2. The messenger RNA is translated into protein. Transcription. The double-stranded DNA untwists. 1 strand of the DNA acts as a template for formation of messenger RNA after RNA polymerase has joined. Complementary RNA nucleotides bind to the exposed strand. Uracil binds to adenine. Guanine binds to cytosine. This results in the synthesis of messenger RNA and the existing DNA rewinds. Messenger RNA then moves through the nuclear pore into the cytoplasm. Translation. In translation, messenger RNA is converted into protein. 3 important components are involved. 1. Messenger RNA from transcription, which carries a series of 3 base sequences called codons. 2. Transfer RNA, which carries a 3 base anticodon sequence complementary to codons on messenger RNA. It also has an amino acid binding site, where an amino acid has bound using energy from ATP. 3. A ribosome, which has 2 transfer RNA binding sites. One site binds transfer RNA with a growing polypeptide, and the other for the transfer RNA carrying the next amino acid in the sequence. Messenger RNA binds to a ribosome. Transfer RNA carrying an amino acid diffuses towards the ribosome and attaches to messenger RNA by its complementary anticodon. The next transfer RNA diffuses towards the ribosome and attaches. The bond between transfer RNA and the amino acid breaks, and a peptide bond forms between adjacent amino acids catalyzed by peptidal transferase. The amino acid chain elongates as one transfer RNA moves away from the ribosome and the next transfer RNA attaches. Polysomes. Usually there are a number of ribosomes attached to a single messenger RNA molecule, all performing translation simultaneously. Such structures are called polysomes, and they ensure that messenger RNA is read at its maximum speed.